Now let's come back to where we left previously. So basically we were going through different molecules which are important for human beings and also other organisms, and some of the important molecules or building blocks are actually proteins, carbohydrates, lipids, and nucleic acids. So these polymers, when the building block of all these polymers are actually monomers, means there is a one monomer which then combines to become a polymer. For example, in the case of a nucleic acid, the single base is a one monomer, which actually we call it a nucleotide, and then when they combine, they make a nucleic acid, either DNA or RNA. And these, there are different definitions, but when there are molecules which are now, they have more monomers, we call them a macromolecules because their sizes are larger. Then all these complex macro molecules which contains the carbohydrates, proteins, or nucleic acid, they also have monomers, as I previously mentioned. For example, in the case of a protein, we have amino acids. The different amino acids actually combine together to make one single protein. And the amino acids define actually the function of a protein. So then how we can produce quite a few different kind of proteins, because everything starts from our sequence which we have in our DNA. Let's say there's a sequence in gene in a DNA which need to be presented, which says maybe develop more of this specific protein which can maybe help the cell to grow, for example. So in that case, the information is copied from a DNA in the form of messenger RNA and then transferred outside the nucleus to ribosomes. And then ribosome is a place where the messenger RNA, based on the sequence of messenger RNA, single amino acids are produced. And those amino acids are then combined together and they are folded into a very complex structure which protein has. And that protein then have specific functions, and those functions are dependent on the messenger RNA sequence or the DNA sequence. Okay, so the code for the proteins is actually present in the DNA, and based on that code, then proteins are synthesized in the factories, which are ribosomes. Okay. So then we also have monosaccharide, which is a sugar to make a carbohydrate, and then nucleotide to make a nucleic acid. There are quite a few functional groups which are very important when we think about functionalizing any nanoparticle that can go inside a body and target cancer cells, for example. There were a few presentations about it, or if it can be a functional group which may be used to functionalize certain surface for diagnostics purpose or therapeutics purpose. So it can be quite a few different forms of functionalization. So some surfaces are easier to functionalize one way or other. So those some of the functional groups are very important because they give some specific properties to the product. And those functional groups are also important when we talk about the molecular nature of our life or macromolecules. So we'll go through some of the important, there are other also available, but some of the important commonly seen functional groups which can be used in one way or other. So for example, first one is alcohols, hydroxide, hydroxyl, which is OH group, which is very important. So OH group is very commonly found in power molecules also, and also when you functionalize your surfaces, you will somewhere see OH group, which is oxygen and hydrogen, which is actually hydrophilic. So alcohols, all the like methanol, ethanol, all has OH at the end, like CH3OH, CH2, CH5OH, so no, C2H5OH, and those are, the OH is a functional group, then R is the external group which you are attaching. So for example, if we attach C2H5 with OH, it becomes ethanol. And if you combine CH3OH, it will become methanol, for example. So the OH is the group which is giving properties to the carbon group. For For example, this is just a CNH, right, which is a hydrocarbon. Itself, CH is a hydrophobic, and because the carbon and hydrogen and all of the living material, they have a carbon hydrogen backbone as a chain. So, but the functional groups comes from the OH group that make it a methanol or ethanol or other materials. Then we also have aldehyde. So in aldehyde, instead of OH, we have a CHO. That is a functional group. So C has a double bond with the O and H. Now the R can be replaced. So acetaldehyde is, for example, one example. So you can have a C2H5 here also. So those functions, this is a basic chemistry which you might have learned also in your high school, but we're just going through some revision, I would say, to refresh your memory. Then we also have a ketones where you have a CO only, and then carbon should have 4 external electrons which can be shared. So you are sharing 2 with the oxygen, and then 2 functional groups can be attached. Let's say in this case, if we attach CH3 on both sides, then it becomes acetone, okay, which is a very important chemical used very commonly in the labs for different purposes. So then it becomes CH3, CO, CH3, right? Then carboxylic acids, very important. When we talk about amino acids, then you will see that carboxylic acid will also come into play, but we'll talk about it because amino acids are the building block of what? Amino acids are building blocks for proteins. Yes. So proteins. So it means in amino acids, the most important, one of the important functional groups is the carboxyls, right? So if amino acid is a carboxyls, it means carboxyl is involved in all of those proteins which we have, and our body is made up of a lot of proteins. How much proteins? Let me see. In percentage, I think it's about 60 to 70%, right? Our proteins. And then there's a amino group, which is NH2, very important. Amino, almost all the proteins have amine group, which is NH2. So it means if you want to attach proteins with the surface, you have to functionalize the surface with something which can bind with amine group, for example, which is NH2. But in this case, NH2, if you attach some functional group, let's say CH3, which is a methyl, so it becomes methylamine. So when we talk about the amino acids, I will not tell you right now, but the amino acids also have a NH2, and then also on the other side it has a carboxylic, and then in the middle you can attach then different functional groups. The rest of the structure stays the same, but only the functional group will be defining different amino acids, and we'll go through that later on, but right now just stick with the amine only. Okay, so when you have NH2 and anything can bind with it, right? So because nitrogen should have 3 external electrons, so it will share 1, 2, and third bond, and carbon has 4, so it's pretty fine and stable in this format. So, and then we also have a phosphate, which is also a very important functional group. You know why? Because I didn't talk about it previously. Where phosphate is involved? Phospholipid membranes. Yes, we have a phosphate there, but I was actually in my mind, I had another thing. What? DNA or RNA. So every base of a DNA or RNA has a phosphate group there. And so it is very important. And that brings the charge. If you look at it, it has a charge minus 2, right? So, and that charge is there. That's why DNA actually becomes charged. And it becomes negatively charged because of a phosphate group which is involved in making the DNA. So one single base of a DNA. But in the phosphate, it can attach with the sugar group, it can attach with the other glycerates, and then it makes different functional groups, for example. And then we have sulfihydryls, which is SH group, which is very important. Anything which has a SH group, which is thiol. Do you know? But let me first finish that. So if anything has a thiol, and you can also have some functional groups with it, which will make different compounds. But for thiol group, anything which is a thiol, what material it likes to bind? Gold. not nanoparticles, gold. So gold and thiol actually show very good adhesion. So anything, if you have gold on a surface, if you have electrodes, it can be in the form of nanoparticles. The gold shows a very good attraction towards the thiol. So whenever we want to functionalize any surface which is made up of a gold, which is very commonly used surface chemistry, gold nanoparticles have been thoroughly investigated for different purposes, also for drug delivery, as a carrier to deliver drug inside the body, and also as a contrast agent. So they are very important material, and how you can functionalize it with the different. So anything which need to be added with the gold, the one way is that thing should be having thiol group, then it will bind with gold automatically. And it shows a very good bonding, very strong bonding. So these are some of the groups which are important, plays a role. I was not sure whether we went through these slides or not. Did we? No? This one. So, okay, but we'll go through it quickly. So inside the cell, I told you that we have a nucleus. We also have ribosomes. Ribosome is a place where, I would say, it's a factory, a manufacturing unit of our cell to make proteins based on the sequence, which is based on the code which is present inside the DNA. So let's say if there is a specific gene, we need to be expressed. When you say expressed, it means the protein will be synthesized to perform different function. And those proteins will be synthesized based on the sequence of a DNA. Okay? And that is done in ribosomes. In ribosomes, proteins are synthesized. And these ribosomes are present on endoplasmic reticulum, but there are some of them are also freely moving also. So endoplasmic reticulum is a curly membrane which you have seen in the cell previously in some of the videos also. And these ribosomes are present on top of that. And then the endoplasmic reticulum, there are two types. One is rough, one is smooth. In the smooth endoplasmic reticulum, they don't have the in a smooth endoplasmic reticulum, they don't have the ribosomes. So it means they are not involved in protein synthesis, but they are involved in functionalization of the protein and folding of the protein. So the protein will be synthesized only in ribosomes, but then the smooth endoplasmic reticulum also have some function for glycosylation of the protein and functionalization or folding of the protein, but the proteins are not synthesized there. This is just one example to show you that how the endoplasmic reticulum looks like. So for example, this one, if you see the inner close to the nucleus, there is a endoplasmic reticulum, but it has small dots on the surface, and those dots are ribosomes. Those dots where the proteins are synthesized, but there are also some smooth endoplasmic reticulum, so which also looks smoother. This is a real image, but it doesn't have endoplasmic reticulum, but it doesn't have the ribosomes, but they are involved also in protein folding or functionalization, but the proteins are not synthesized there. So one important organelle of cell, which is considered the largest organelle of a cell, is nucleus. So because why it's important, that the reason is it has all the information, a code for the cell, like how it will perform and what kind of genes are present and how they will be expressed, right? And there are 23 chromosomes present in a pair, so total there are 46 chromosomes inside each cell, and 22 pairs are actually autosomes, and then the one pair is a sex chromosome, which is a little bit different. Means sex, it can be X and Y or both X. So but the point is, out of 46 there are 22 pairs which are non-sex related and 2 are sex related chromosomes. But when talk about the chromosome, it is made up of a DNA. But if it is made up of a DNA, that does it look like a DNA, this structure? No. So this X which you see here, the arm, which we call it as a chromatids, they have DNA very well packed with a protein called histone. So the DNA is very well woven in a certain form around the protein, and then it's very well packed structure, and then that is made up of then that makes a chromatid, and then makes the whole chromosome, and there are quite a few of them, like 23 pairs. So basically, whenever there is some information need to be sent out, then the messenger RNA, actually there is an RNA, I think it's called the RNA, riboxynucleic acid, and then that RNA actually copies some information from the DNA, maybe from this part, for example, copies information, complementary, and then take it out through the nucleus pores, right, to the endoplasmic reticulum, where there are ribosomes which will make proteins, and then the proteins then perform different functions. They may go outside of a cell, or they may stay inside of a cell based on whatever function they want to perform. Okay, this is how the nucleus looks like. So the nucleus has a small pores, and the pores are important to transfer mRNA from inside to outside, right? Correct? So, and then this is from where the information is taken out from inside the DNA. And then these pores can also be used now, if let's say if there is a gene which is causing a disease in a human body or disease in a cell. So what we can do is gene therapy, right? So we can go, if we know that this is an area where this gene is not good, so we can knock out that gene, right? Theoretically. So we can knock out that gene and maybe replace that gene with something which is not mutated, means something which is a good gene. So, and then we can change the cell functions, because then cell will function according to the DNA. So if we know the DNA, then if we can change it, we can change the function of the cells, and then we can control the fate of a cell to some extent. Okay, so let's talk about more specifically about what is the nucleic acid first. Again, how it's made, because either it's a ribonucleic acid or deoxyribonucleic acid, which is when we say DNA is a deoxyribonucleic acid, one thing is acid, then there's a deoxyribo, there's another thing which is called deoxyribose. There's actually a sugar group which is attached, and we'll show you why it's called deoxyribonucleic acid and why it's called ribonucleic acid only. That's the only difference. Otherwise, their structure is very similar. And definitely it's a polymer. So DNA is a polymer. So polymer, it means it has a different monomers, and each monomer will be called nucleotide. So single base, sometimes we call it a base, like the basis of a single base, which is A or T or G or C, or sometimes we also call it as a nucleotide. Okay, so it's the same thing. Okay, now the nucleotides, or each base of a DNA, consists of 3 different sub-units, and that makes a single base. So one is a pentose sugar. That's a sugar group, which is formed in case of a 3 in a structure of a pentose, which is 5 corners. 1, 2, 3, 4, 5, right? Okay, 1, 2, 3, 4, 5. And that's a sugar. Then we also have one phosphate group, I told you before. So the DNA and RNA has a phosphate groups, right? And that brings the charge. And then we also have a nitrogen containing base, which we didn't mention before, but there is also base which give a functional properties, whether it's a, whether it's a T, whether it's a G, or whether it's a C. So basically, the sugar group and phosphate group is the same in all bases. Only the difference is actually the nitrogen containing base functional group that would be defining these final properties of a nucleotide. Okay, now let's look at first a sugar group, which is present in the structure of a pentose sugar. So this is for ribose. This is called the ribose sugar group. This is deoxyribose. Do you see any difference between these two? Yeah, H versus OH. So there is no O here, right? But there is a O here, correct? So when there is no O, we can call it deoxy, right? So no oxygen. So deoxy. Other thing is the same. So that's one major difference between DNA and RNA. That the sugar group which is used in a backbone, it doesn't have an O here, but in an RNA, there is a ribose sugar which has an O. That's it. So now the carbon has a one H, then one is attached with the carbon, so second, and then there is a OH on the other side. In this case, instead of OH, there is a H only. So that gives it a name. So we can deoxyribose. So we kind of, now the nucleic acid is missing here. If we attach a nucleic acid here, it will become DNA. And same is true with the ribose nucleic acid, okay? So let's start adding now the nucleic acid there. So this is how the DNA looks like. So basically, this was our sugar group, which is the orange here, which we looked previously, the pentose sugar. Then the sugar group can be added with the phosphate, right? I told you the phosphate group is there in a sugar, in a DNA. So the sugar group actually binds with the phosphate, and I'll show you later on that where it binds, but it binds with the, sugar binds with the phosphate. So these two units are always present, whether it's A or T or G or C bases. Now, sugar plus phosphate combined with some base. So in this case, it's a guanine, which is G. So once it's bind with the G, then it makes one nucleotide. So this is a G nucleotide. So if you look at the DNA structure in total, so it's a double helix, right? This is a double ladder, not a helix. So actually, in reality, it's also curved, but this is showing a simplified version. There you have a different G's and C's, and you can see one thing which is common is a sugar phosphate. There is a sugar, orange. It is a phosphate on one side, phosphate on the other side. So phosphate groups are actually keeping those DNA together. So that's kind of a building block of the backbone. So if you remove the phosphate group, the bases cannot stay together, correct? So if we want to disintegrate the DNA, what we can do is we can somehow do something to the phosphate. Maybe remove it or somehow react it with some chemicals. So if we remove the phosphate, it means the DNA would not be present in a full form. It will become single bases. Another thing which is important is the bases which will make things different. So which is the G's and GC, all the G. Guanine always goes with the C, and then adenine goes with the T, thymine. So those are 4 different bases which we have, and those are called in nitrogenous bases. They also have some chemical structure I'll show you in a minute. So adenine, thymine, these are called the porines, and the pyridamines are cytosine and thymine. So briefly, we'll say it also C and T, A and G, right? So A binds with the T and G binds with the C. But do you see any difference between GC and AT in this figure? The bonds. Do you see there are 2 lines here and 3 lines in GC? So that shows actually hydrogen bonds. Okay, so it's not a covalent bonding. So you know what is hydrogen bonding, by the way? Do we see hydrogen bonding any other place? Water, right? Water, H2O. So the oxygen has 2 H. So how many electrons oxygen has in the outer shell? How many single oxygen has electrons in its outer shell? 6, because it's from group 6. So it has a 6. So if it needs 2 more, right? If it needs 2 more, then it becomes stable. And otherwise, single oxygen is very reactive. And that's why we always found it in the environment in O2 form, right? Of O2. So it shares 2 electrons with another oxygen to make a molecule, and that is not reactive. Because otherwise we cannot survive. So, but the point is, now the 2 hydrogen atoms, oxygen and 2 hydrogen, make up 8, and the hydrogen also need 2. So it has 1 and 1 shared, so it also becomes 2. So it's covalent bonding, right? So then why we get a charge actually on a H2O molecule? So if you remember, when one atom is larger, it I think pushes the electrons a little bit away from it, right? And that gives a partial charge to the structure. So I don't remember which has a partial positive. I think oxygen might have a partial positive and the hydrogen has a partial negative, but I'm not sure about that. So that structure, because two atoms are different sizes, and the atoms which are shared, the electrons which are shared, they are pushed one way, a little bit, the shared ones. This is a simple version how to explain it, and then that gives a partial charge. Otherwise, if it's everything is like a hydrocarbon, carbon, and 4 hydrogen atoms, they are pretty stable. But in this case, we see some partial charge on the surface. So partial positive, partial negative. So it means the H2O single molecule has some partial charge. And now if you put all those molecules together, so you will see a bonding between hydrogen and other oxygen because of a charge, right? So there are H2O, H2O. Now the one H from another molecule actually shows some attraction towards O from another molecule. So that shows the hydrogen bonding. So basically, when I say hydrogen bonding, it's the oxygen. No, hydrogen bonding is the hydrogen showing bonding towards oxygen, nitrogen, and phosphorus. And oxygen, nitrogen, phosphorus, hydrogen shows bonding towards that. That's called the hydrogen bonding. So in this case, we have 3 bonds of hydrogen. We have 2, and I will show you in a minute why we have 3 in one case and 2 on the other. Okay. So, and this has 2 hydrogen bonds. So now the question is for you guys that which one would be bond, which one are actually conjugated more strongly? The ones with the 3 bonds or 2? 3 for sure. So it means if we later on want to break this double strand into 2 single strands, right? So if let's say it was all G's and C's, GC, GC, GC and all that, then it would be harder actually to break them, right? Because they're strongly bound with each other. But if it was all A's and T's, then it will be comparatively weaker. So by the way, the DNA, when we break it from double-stranded to single-stranded DNA, it's a one strand. So we say double-stranded to single-stranded DNA, then we call it a DNA melting. We melt it from double to single. Okay, that process is called DNA melting. So when we melt the DNA, so there is also melting temperature, and the melting temperature depends on hydrogen bonding. Now, if there is some DNA sequence which has a more GCs, it will have more melting temperature or less melting temperature. More melting temperature. It means you really have to heat it up to a certain level before it can break into 2 single strands. So it means your melting temperature depends on the length of your DNA also, and also the composition, whether it's a G's and C's or A and T's. Okay, but let's move on. So now we have actually a ribose or a deoxyribose, which is your sugar, pentose sugar. It combines with the phosphate, I will show in a minute, but then it also combines with the base. So the bases were cytosine, thiamine, CT, and then A and G, which is adenine and guanine. So these are how the bases are formed. It's also based on CH, and nitrogen is there. And there is amine also there in H2. So those bases define whether it's a A-type nucleotide or B-type or C-type. But there is a fifth actually base also there, which is called uracil. Okay, but in DNA we have not seen U so far, right? But U is actually present in the form of when we talk about the RNA. In RNA, okay, what is the difference between these 2 bases, thiamine and uracil? CH3 is missing only there, right? Otherwise, it's the same thing. So what happens when in the second important difference between the DNA and RNA, that in RNA we don't have a thymine, we have a uracil, okay, which are similar in structure, but they are different. So it means if we make our own new base, very similar to this one, let's say if I knock one hydrogen and maybe attach some other functional groups there at this point, so it means I can synthesize my own new type of nucleotide, right? So instead of A, T, and G, C, which is naturally present, I can have some new types also. Correct? So it means we can have some DNA which is different than ATGC thing. It may also combine with its counterpart. There is a company. It's a startup company. It's called the Firebird, based on I think some professor, very well-known professor from UF. Now he's retired and running this company. It's called the what is it? Firebird, right? Let me see the name. Oh, it's called the Firebird Biomolecular Sciences. So if you look at this company and let me see what products they have. So they have actually designed some bases which are slightly different than the regular bases, and we can do many different things. Let's say in this case there is only one H bond instead of two, for example. So you can look more into it. So this is some very latest research, and they have quite a few applications in diagnostics and other areas. But I'll leave you with that note only, that we can play with these bases ideally, and we can generate new bases also. But in natural environment we have a sugar group attached with the base, any of those, and then there is also phosphate group attached. So the phosphate, sugar, and base. So base is changing. This thing stays the same. That will define one single strand of DNA or RNA. So before going further into details of the structure, I want to actually show you first a video today, and then we'll come back to the slides which I skipped about the DNA structure. I think I gave you a little bit of information, which is fine, and then we will talk about this new topic briefly, and I want to show you actually a video, give you some brief background about the video. And then in the next lecture, I will go through the structure of a DNA, the remaining slides, which I skipped once again. So that will also make sure I'll repeat some information so that it may stay there in your memory for a little bit longer time. Okay, so because those are important. So those basic structures are very, very important for defining later on the devices and technologies which you will be building based on those structures. Like nanopores, the other DNA sequencing methods, they rely a lot on structure, how DNA is made, and that's important. So, okay, now there was one project which actually started in I think 1997, I believe. It's called the Human Genome Project. We also call it a HGP. So, the Human Genome Project was that before that time, we have not sequenced the whole genome of human beings. Although people know about some genes, but no one has the information, the complete sequence of human genes. And why it's important? Because we want to know, okay, how all the DNA looks like, and maybe if we have DNA from 1,000 people, we can see that how it's different from one another, right? And it can be very helpful later on when we want to compare. If someone has a disease, we can compare, sequence his DNA or her DNA and compare it with these DNAs which are sitting there, right? And it can be used for a lot as a database for a lot of research later on. So that was the goal, to sequence the whole DNA, which involves approximately 30,000 genes. So what is a gene? So DNA is long. Some section of a DNA might perform some specific function. So that specific segment of a complete DNA is called the gene. Okay? Because not a single base can provide you that function. So it has to be multiple bases that can maybe defining some specific function is called the gene. So there are more than 30,000 genes, and there are 37 genes in the mitochondrial genomes. There are also some DNA in mitochondria also, which is smaller than the main DNA nucleus, cell nucleus DNA, but it is there. So the goal was actually to identify all the genes in human DNA, determine the sequence of the 3 billion base pairs, which is like sequencing the whole DNA, improve the tools for data analysis, address the ethical issues, transfer related technologies to private sector, and there are quite a few, but major objective was actually to sequence the whole human being DNA. But okay, this started in 1990 or 97, so it took 13 years to complete it, and was mainly led by Department of Energy and NIH. It was planned for 15 years, but it took actually, it was finished in 13 years, faster than what it was planned for. It happens very rarely. Normally, whatever you plan, we normally take longer time, but in this case, it was actually finished earlier. And now I'll show you a video, which you can also run it when I show the folder, the PowerPoint with you later on the canvas. It doesn't run on a Mac because I was trying to run it on a Mac, so it didn't work. So you should use a Windows if you want to run it at home. Okay, the video. It shows you, so you may not understand some parts of it if you are not from biology background, but it will give you some understanding how the DNA was sequenced. Because sequencing means we want to know the what type of DNA bases are there, like whether it's AAA, TTT. So what is the sequence? Because there will be only 4 bases, but how they're arranged, and that defines their functions. So, but there was no technology out there at that time which can be used for DNA sequencing. There were some technologies which were very crude, and it took longer, and that's where they will explain you how the DNA was sequenced. But nowadays, actually there are other methods which we'll also discuss later on during this course, that what are other ways to sequence the DNA. And now it's much cheaper, actually much cheaper and faster than it's done like this in this video. Okay, so if the whole project has to run again, maybe they might finish it in 1 year. At that time, because of the focus of sequencing the whole DNA, there are also a lot of effort to actually develop new technologies how we can efficiently sequence the DNA, and that's why the technology actually progressed, and we were able to get better DNA sequencing devices. So now, before I spend more time on talking, I think let's run the video, but for that I have to switch the computer to room PC. Okay. Just double click on that, it'll run.
Studying the human genome, the complete set of human genes, is a way of studying fundamental details about ourselves. A 4-letter alphabet makes up the 3 billion letter-long sequence of DNA that, divided into 23 pairs of chromosomes, inhabits the core of each cell in our bodies. Of course, people are not identical, and DNA sequences do differ subtly between individuals. The Human Genome Project is producing a representative sequence. Separate projects are charting variations in the sequence. The representative sequence is a composite from several people who donated blood samples. Originally, close to 100 people volunteered to give blood and gave their informed consent. affirming that they agreed to the study of their DNA. No names were attached to the blood samples, and ultimately scientists kept only a few. These measures ensured that the DNA sequence would remain anonymous. Not even the donors knew whether their samples were actually used or not. The Human Genome Project aims to read, letter by letter, the 3 billion units of human DNA. The Human Genome Project scientists began large-scale DNA sequencing in 1999. Before starting to sequence the human genome, they built maps of the human chromosomes and developed and refined techniques for analyzing DNA. With the tools in place, genome scientists took just a year to amass sequence covering more than 80% of the genome. The human genome is a massive text. If the 3 billion letters, called bases, that make up all the human DNA in a cell were printed in telephone books, the stack containing the whole genome would reach as high as the Washington Monument. To figure out the sequence of all the bases in the genome accurately, scientists need to read the 3 billion bases not just once, but at least 6 to 10 times. Sequencing reactions can only reveal the order of a few hundred letters of DNA at a time, amounting to a fraction of a page. Having many overlapping segments of sequence allows the genome to be puzzled back together into an intact whole. Before shredding the DNA in the genome and starting to sequence, Human Genome Project researchers first built a map of the genome. They found thousands of landmarks scattered throughout the chromosomes to help them navigate among all the DNA. Developing genome maps was a key step to prepare for DNA sequencing, but the increasingly detailed maps have also been an important tool orienting hundreds of geneticists hunting for disease genes. With enough markers in place, the Human Genome Project scientists created libraries of clones that span the genome. Each of the clones contains a manageably small fragment of human DNA that is stored in bacteria. Scientists can tell what part of the human genome each clone derives from by figuring out what markers each contains. This clone-by-clone approach for analyzing the human genome makes it possible to double-check the locations of sequence. And because the Human Genome Project has been an international effort with many laboratories taking part, carving up the genome has allowed different groups to coordinate their work effectively. Clone libraries offer the advantage of real libraries, orderly access to information. In most clone libraries, fragments of human DNA are stored in a kind of bacterium, E. coli, that normally lives in our large intestines. Each E. coli cell in a library stores a single segment of human DNA, so that the human fragment can be tracked and copied easily. To sequence the human genome, Human Genome Project scientists cut relatively large clones called BACs, which are 100,000 to 200,000 bases long, into smaller fragments. The smaller fragments, which are about 2,000 bases long, are typically stored in E. coli viruses. The scientists determined the precise order of the larger clones because pinpointing the positions of many smaller clones is much more work. But for actual sequencing reactions, the smaller clones are more suitable. E. coli cells containing fragments of human DNA can be stored in freezers indefinitely. When researchers are ready to retrieve DNA from the library, they revive the cells by bringing them back up to 37 degrees centigrade, gut temperature. To make many copies of the human DNA, the E. coli cells act as copiers. A few related cells containing the same bit of human DNA inside them are released into a rich, warm broth. Machines shake the broth vigorously, so the cells have plenty of air and divide rapidly, about once every half hour. After a single night, a third of a teaspoon of broth contains billions of copies of E. coli. And so, billions of copies of the particular fragment of human DNA they contain. The next morning, the cells are broken up to release the DNA inside. The DNA is separated from the cell debris and washed clean. Now there are enough clean copies of the segment of human DNA to set up a sequencing reaction. A sequencing reaction includes 4 main ingredients: template DNA, copied by the bacteria; free bases, the building blocks of DNA that come in 4 types; short pieces of DNA, called primers; and DNA polymerase, the enzyme that copies DNA. The chemical reaction that makes DNA in a test tube is very similar to what happens in a living cell. Both rely on DNA polymerase, and in both cases, DNA strands have a head end, which scientists call the 5' end, and a tail end called the 3' end. A DNA strand can grow only from its 3' end. Making DNA in cells and sequencing DNA in test tubes depend on one central property of DNA. The building blocks on opposite strands of DNA pair specifically. A C always pairs with a G, and an A always pairs with a T. The primer alights on the segment of DNA that matches it. Free bases that match the template sequence can attach to the new strand's growing 3' end. Among all the free bases swimming in the solution, a few have an extra chemical part. The chemical is a fluorescent dye. When the colored bases attach to the growing strand, the extra chemical part keeps the new DNA strand from growing any further. A different colored dye is attached to each of the 4 kinds of bases. A completed sequencing reaction contains an array of colored DNA fragments. The shortest are the length of the primer plus 1 colored base. The longest fragments are usually between 500 and 800 bases long, which is when the sequencing reaction runs out of steam. The products of sequencing reactions are fed into an automated sequencing machine. Sequencing machines have become increasingly sophisticated over the last decade, running more samples, processing them more quickly, and requiring much less labor to set up. The DNA molecules produced in the sequencing reaction are separated by a process called electrophoresis. DNA molecules are negatively charged. The sequencing machine sets up an electric field. All the DNA moves down through a porous gel toward the positive charge. Shorter fragments of DNA move more quickly through the holes of the gel than larger fragments do. In the sequencing machine, a laser excites the fluorescent dyes, and a camera detects the lights that the excited dyes emit. One by one, the sequencing machine reads the DNA molecules passing down the gel and sends the information to a computer. A single sequencing reaction reveals the sequence of a few hundred letters of DNA. A computer program helps integrate the information from individual sequencing reactions. It spots where fragments overlap to puzzle the pieces back together. Many overlapping sequencing reads are needed to reveal the uninterrupted sequence of the original stretch of DNA. On average, every base pair of human DNA will be sequenced 9 times. Some stretches of DNA are easier to read and need to be sequenced a little less often to get high-quality sequence. Some stretches need to be analyzed more exhaustively to get finished high-quality sequence. To sequence the human genome, scientists will ultimately run more than 50 million reactions. Some 2,000 scientists in more than 24 labs around the world have worked toward the goal. The Human Genome Project scientists have agreed that whenever they assemble a stretch of DNA that spans 2,000 or more letters, they will send the data within 24 hours to public databases. Anyone with access to the internet can then see and analyze the sequence. In the spring of 2000, after sequencing the 3 billion letters in the human genome an average of 4 times, the Human Genome Project had released DNA sequence for 90% of the human genome. This working draft sequence is 99.9% accurate. Some gaps and ambiguities will remain in the representative genome sequence until each letter of DNA has been sequenced approximately 9 times. The working draft has just half of that information, so it contains gaps where sometimes just by chance sequence was not obtained for particular regions. Sometimes the chemical properties of some stretches of DNA make particular parts of the genome harder to capture and analyze. There are also many repeated sequences in the human genome that complicate assembling the complete genome sequence accurately. Some repeats are short, some are long, some are present in 1 million copies, others are repeated only twice. Before the human genome sequence is considered finished, scientists must resolve all ambiguities that can be resolved, and one by one close all gaps that can be closed with modern sequencing technology. Ultimately, there will be no more than 1 error per 10,000 bases. In other words, the sequence will be 99.99% accurate. The finished human genome sequence is expected by 2003.
So try to watch it one more time when you go home, okay? Um,
You
and
will publish the link.
yeah, I will attach it. Yeah, I will upload it on Canvas. Yeah,
Or the link.
The link, it's actually a file which when you double click it, it's an exe file and it runs it on your computer.
Okay.
This is how it works. There is no link for it. This is just an exe file, but make sure that you run it on your Windows. It doesn't work on a Mac, that specific file, but there may be a Mac version which I don't have it. Okay, see you guys on next Tuesday.